Hello, my name is Mark Lennox. I'm a computer science PhD student at Queen's University Belfast. I'm here to give a presentation on my paper titled Deep Phosphosite. In this work, we utilize a transformer model for predicting general and kinase-specific phosphorylation sites within a protein. Deep learning has transformed sequence analysis with the introduction of the transformer model in 2017. However, this new form of deep learning has yet to be fully utilized in a biological setting. Our aim is to use both a set of convolutional layers and a transformer to detect phosphorylation sites in a protein. In doing so, our approach will be able to identify critical k-mers and accurately model the long-range dependencies within a phosphorylation site. Since we are not using recurrent neural networks, the output of our model can also be calculated in a parallel setting and should be able to reduce the time required for training. The phosphorylation site data used in this investigation was based on the original data collected in the Music Deep paper, which was obtained from the UniProt database. In both the general and kinase-specific investigation, we used the most recent version of the PhosphoSite Plus dataset to update the labels from the previous work. For a fair comparison, we also used a window size of 33 amino acids for each of our models. And just like previous studies, we also trained a deep learning model for each of the kinase families. This diagram is an overview of the deep neural network we use to model each of the sites. It has been broken down into 3 key stages. Inspired from past work within the field of protein modeling, we have opted for using an embedding layer along with a stack of convolutional layers. Each convolutional layer is then connected to the previous layer through the use of a residual connection. Such a design will ease training and should avoid the latter layers from overfitting to specific patterns within each site. The encodings produced by the convolutional layers within the first stage of the model are then passed to a 4-layer transformer. These encodings are then finally passed through a set of fully connected layers to produce a final prediction for each site. Unlike the past examples, this model is not trained using binary cross-entropy. Instead, we use focal loss, which penalizes the model for easily classified cases, which would be the majority of the negative site data used during training. As mentioned, the same training and testing sets were used for all of the models. A 5-fold cross-validation was performed to evaluate the performance of deep phosphosite against past examples. As you can see, our model easily outperforms both baselines. And again, for all 5 kinase-specific tasks, we observed an improvement over the baselines, with our model producing the highest area under the curve scores for each kinase. Both of these figures are Tsne plots for the general and residue-specific sites respectively. From the left figure, we can see that the model moderately separates both types of site. However, there is still a significant overlap between the 2 examples. This is perhaps due to the limited window size used when testing, or perhaps it is due to the model lacking information about the complete protein sequence. From the figure on the right, we visualize the same encodings but now with the residue-specific labels. Unsurprisingly, we can see how the network clusters the tyrosine examples together in a way from other sites. These plots, however, outline the significant flaw with just considering a small window size of a protein sequence, and this is something that must be addressed in future approaches. These figures consider the weights of all 8 attention heads when the following site is passed through our model. On the left figure, initially, these heat maps do not reveal any additional information with regards to the critical parts of the site. On the right figure, however, by correlating the weights, we can now see what parts of the sequence actually attend to the site in the middle of the sequence window. These correlations indicate that the right-hand side of the window is more critical to actually classifying the site. In this paper, we present deep phosphosite as a novel method for both general and kinase-specific phosphorylation site predictions. Our approach takes raw sequence data in as input, and without using any other tools to generate features, it can produce a prediction for a given site, and outperforms other well-known tools on benchmark datasets significantly. As an example of white box deep learning, we have also visualized the features generated by our model through the use of Tsne plots and heat maps. In future work, we aim to test pre-trained algorithms for protein modeling, such as a BERT-style model, which has previously shown state-of-the-art performance on many natural language understanding tasks. Thank you for listening.